Hello, my name is Anna Rivas and I'm going to be doing the first video of this course, and that is for Lab 1 of the Endocrine System, where I will be showing you how to digitally label a picture. We're going to be labeling 3 cells found in the pituitary gland, 2 of which are found in the anterior pituitary gland, and those are acidophils and basophils. Then we're also going to be labeling a 3rd cell, which is found in our posterior pituitary gland, and that is actually going to be the nucleus of a pituocyte. Before we actually start labeling the photo, I just want to be able to distinguish them visually first, and in order to do that, I did some research and saw that our acidophil cells are stained red in the histology slide, and our basophil cells are stained blue in the histology side. We also know that since it's found in the anterior pituitary, they're going to be separated from the pituocytes, which are found in the posterior pituitary, and the matrix for those both areas are completely drastic. You can definitely tell which is the posterior, which is the anterior side, and that's going to help us kind of distinguish where the nucleus of the pituocytes are. Now, when I am on a Mac, I like to use the photo editor Pixel Style app that is found on there. But if I am on a desktop, I found that the easiest application to use would be Paint. Way easy, straightforward. So, but for right now, for this video, we're going to be doing it on a Mac. So we are going to be using the photo editor Pixel Style. Perfect, so we have switched it over so you can see the MacBook, so we can start digitally labeling our pituitary gland slide. And just so we can double check, it is the photo editor Pixel Style app found on the MacBook. So to begin, I go to the pencil tool, and then I go over here and make sure that I click to draw a straight line, because I want to have a straight line pointing to the structures. And then I'm going to get closer. And like I said earlier, it is very easy to distinguish where the anterior and the posterior lobes of the pituitary gland are. Anterior lobe has a thicker matrix, and they have more cells filled in the matrix. So that is right here. This is the anterior part of the pituitary gland, my apologies, and this is the posterior of the pituitary gland. Let's start labeling the structure. So to label the structure, again, we're going to go over here, make sure we click on our pencil tool, make sure that if you have a color preference, you pick the color of your preference. Mine is red. Exit it out, and then always make sure that it has the draw straight lines. So I like to click here. I'll click once here, and then I'll click once over here, and it immediately puts a line. And so then I see that there's other ones that are very noticeable. I might want to do another one. So from there, since I've already clicked here, I click to another one that I want, and let's just say this one right over here, which is also very noticeable. Perfect. And so now I want to add some text. I'm going to come down over here to where the T is, which stands for text. If you have a color preference, once again, you can change that. I like red, so I'm going to put red, and then I'm going to cross that out. And then I'm going to click here and start typing in the name of it. And since we did do more than one nucleus, it's going to be nuclei, which is the plural term of the site. Perfect. As you can see, it automatically puts it at font size 40. So now we're going to change that down to font size 12. Makes it significantly smaller, as you can see. And then we're going to move it where we want. So we're going to just reposition the text. Perfect. So next, let's label the acidophil, which is found in the anterior part of the pituitary gland. So the acidophil, once again, is stained red, so it's lighter in appearance. We're going to go ahead and come back here and hit our pencil. I like the color red, so I'm going to hit red again. And I also want to make sure it's on straight lines, which it is. Good. So right over here, there's a very clear little cluster, it looks like, right here of acidophils. Make sure I hit the pen. Perfect. So we're going to go to that cluster. Wonderful. And I'm going to now go back to the text, label it. Once again, I labeled it. Perfect. We're going to now go on to the basophil cell, which are stained the dark blue, found in the anterior lobe of the pituitary gland. We're going to make sure we hit our pencil again. Make sure that our pencil is red or whatever color of your preference. If you don't care if it's orange, keep it orange. If you want it black, put it black. If you want it red, put it red. Here, make sure it's draw straight lines, and it is. Wonderful. Now let's find one of those basophils. So the basophil, you can see it's actually very noticeable. There's a few we can choose from. Let's go ahead and do this one. I'm going to point it out here. Perfect. Say you want to do another one, you could. I'm just going to keep it at the 1. And then I fast forwarded on the last one, but just so we can recap, make sure that we're comfortable with labeling with the text, I will film it. So we're going to go over here. Once again, we're going to hit the T that stands for text. Make sure it's on red or whatever color of your preference. That is my preference. And then I'm going to hit it over here. And sometimes when you change different parts of the picture, it changes the color again. So I'll just once again hit red. And I will go ahead and type it. This is the basophil cell. Perfect. Basophil cell. And then I'm going to once again change the size of the font to 12 or whatever of your preference. I prefer 12. Makes it significantly smaller and fit perfectly in the picture. Move this here. Wonderful. And then you can go ahead and save the picture as you would any other picture, and it becomes a labeled, digitally labeled picture that we can now put into our lab report. Thank you very much for watching my video.